Brooke here. Today this video will be on capillaries, so stay tuned. Okay, getting further along into capillaries. I'm gonna give you a brief overview first. And capillaries are just the smallest type of vessel that we have in our bodies, and they are composed of a single layer of endothelium with a basement membrane. The endothelial layer is simple squamous endothelium, in case you remember that from last semester. And the endothelial cells can exhibit different features depending on where they're at in the body. These can be anything from tight junctions, which is a fusion of the plasma membranes of adjacent cells, intracellular clefts, which include spaces between cells, fenestrations, which are pores in the plasma membrane covered by a small thin membrane, and they can also exhibit vesicles. There are three types of capillaries that we're going to go over today, and they're continuous, discontinuous, and fenestrated. We're going to utilize those features that endothelial cells of capillaries can exhibit and make it make sense with the three types of capillaries that we have. Okay, so first off, I'm going to show you a couple pictures. These, 1 and 2. So 1 is an intracellular cleft. You can see those small spaces between the cells. And 2 are vesicles. So vesicles are involved in transcytosis to remove molecules from the cell. Next off, we have labeled 3 are tight junctions, and if you remember, those are the fusion of plasma membranes of adjacent cells. So that means their plasma membranes are fused. Okay, and 4, we have the fenestrations, and these are just pores in the plasma membrane covered by the small diaphragm or the thin membrane. Okay, so now that you have a visual representation of the features of endothelial cells, we can use those to make the other capillaries, or the types of capillaries, make sense. So first we have the continuous capillaries. These are continuous endothelial cells, and what you can see, they have intracellular clefts, but they don't have any of the tight junctions or fenestrations. And these can be found in the blood-brain barrier, and we'll talk about why in a minute. Okay, then we have discontinuous capillaries, which have the intracellular clefts, and they also have large openings within the endothelial layer, as well as the basement membrane. And those are found in liver. Okay, and this one, we have the fenestrated capillaries. As you can see, the endothelial cell layer is covered with fenestration. Fenestrations. And these can be found in the kidneys and the intestines. Okay, so now we're going to talk about a little bit about why the different features of the endothelial cells matter. And the most important thing to remember is they are important because of transport. So the size of the molecule determines whether or not it can travel through the endothelium, and the features that are present on the endothelium plays a role in that as well. So for tight junctions, you can only have substances that are lipid soluble or have membranes can pass through the tight junctions. But in intracellular clefts and fenestrations, you can have larger molecules pass through. Okay, and then for vesicles, you can molecules can be transported across capillary walls. That's called transcytosis. So the size of the molecule determines whether it can travel through the endothelium. Now, why would this be important in the types of capillaries? Well, if you have large fenestrations in the kidneys and intestines, you need those large molecules to be able to flow through that capillary wall. And the same thought process can be applied to the other two. types of capillaries as well. Okay, that's all I have for you guys. Thanks.